Hello, my name is Alexandra Nunez. It is May 6th, and I am going to be sharing my presentation on the anatomy and physiology of the retina. First, I'm going to start with what vision is and how light enters the eye and how the retina plays a role in converting that signal that we see into the visual stimulus that we need to go to the visual cortex of the brain to process it. So I'm going to start with what vision is. So vision is the process through which light reflected from objects in our environment is translated into the mental image. So it's in 3 steps. Light enters the eye and the lens focuses light on the retina. Then the photoreceptors of the retina transduce light energy into an electrical signal. Neural pathways from the retina to the brain process these electric signals into visual images. So as you can see in this image of the eye right here, so the light is going to enter and hit the back of the eye, which is where the retina is located. It will go through the retina and all the different cells inside the retina that process information, going to the optic nerve into the central nervous system. All right, so the anatomy of the retina. So the retina is a light-sensitive lining of the eye that contains photoreceptors, is also crisscrossed with small arteries and veins that radiate out from the optic disc. And the pupil helps control the amount of light that enters the retina. That's another important tool that connects it. So there are 5 types of neurons in the retina. There are photoreceptors, horizontal cells, bipolar cells, amacrine cells, and ganglion cells. The backing of the retina is the dark pigment epithelial layer. Its function is to absorb any light rays that escape from the photoreceptors, preventing distracting light from reflecting inside the eye and distorting the visual image. As you can see in this image as well, how light will enter, hit the back, and go all the way up through the ganglion cells to the optic nerve. All right, now I'm going to go more in depth in the 5 types of neurons in the retinal layer. So I'm going to start with the photoreceptors. So there's 2 different types. There are rods and cones. So rods are sensitive to light levels, which help us see in low lighting. Cones are responsible for high acuity vision and also our color vision. Sensory information about light passes from photoreceptors to bipolar neurons, and then a layer of ganglion cells, which are neurons of the eye whose axons form the optic nerve. There's also horizontal cells and amacrine cells. So horizontal cells synapse with photoreceptors and bipolar cells to mediate lateral inhibition of the retina. So lateral inhibition of the retina, it enhances stimulus location and contrast. And amacrine cells modulate information flowing between bipolar cells and ganglion cells. All right, so then I'm going to talk about how photoreceptors perceive vision and how that signal will then pass through the bipolar cells and ganglion cells. Starting with this, so light-sensitive visual pigments are bound to a disc membrane in the outer segments of the photoreceptors. These transducers are that convert light energy into a change in membrane potential. So the 2 different types of visual pigments for the rods, there is rhodopepsin, and then the visual pigment of the cones are actually it's red, green, and blue cones. The visual pigment of cones are excited by different wavelengths of light, allowing us to see in color. And each cone type is stimulated by a range of wavelengths, but is most sensitive to the particular wavelength that it corresponds with. Our brain recognizes color of an object by interpreting the combination of signals coming to it from the 3 different color cones. And then talking about phototransduction, which is in the process of phototransduction is similar in both rhodopepsin and the 3 color pigments of the cones. Kind of how to translate that signal. So signal processing for this in the darkness, rhodopepsin is inactive and CGMP is high, and CNG and potassium channels are open. This allows for a tonic release of neurotransmitters onto bipolar cells. Then the light will bleach the rhodopepsin, opsin decreases the CGMP, closes the CNG channels, and hyperpolarizes the cell, which allows for neurotransmitters released to decrease in proportion to the amount of light that is hitting the rods. And in the recovery phase, the retinol recombines with the opsin. All right, and in signal processing, a glutamate release from photoreceptors onto bipolar neurons begins the signaling process. Um, so there are two types of bipolar cells. There are the on and off bipolar cells. The on bipolar cells are activated by light when glutamate secretion by photoreceptors decreases. In the dark, the on bipolar cells are inhibited by this glutamate release. Off bipolar cells are excited by glutamate release in the dark and is inhibited when there is less glutamate in the light. Bipolar cell signaling process also is modified by input from horizontal and amacrine cells. And each ganglion cell receives information from a particular area of the retina. So these are called the visual receptive fields. There are two types of ganglion cell receptive fields. There is an on-center and an off-center field that is associated with the ganglion cells that responds most strongly when light is brightest in the center of the field. If light is the brightest in the off-center region of the field, the on-center / off-center field ganglion cell is inhibited and stops firing action potentials. And then the reverse will happen with an off-center / on-center field. There are multiple types of ganglion cells in the primary region of the retina. There are M and P cells. M cells are sensitive to information about movement, while P cells are sensitive to signals that pertain to form and fine detail, such as texture of objects in the receptive field. And then once action potentials leave the ganglion cell bodies, they'll travel along the optic nerve to the central nervous system for further processing, going to the visual cortex in the optic nerve. Then there is a phototopic organization that is maintained in the visual cortex with 6 layers of the neuron grouped into vertical columns. Within each portion of the visual field, information is further sorted by form, color, and movement. So I hope this kind of showed how information is passed and how it goes through the retina to form this signal that the optic nerve will send down the optic tract into the visual cortex of the occipital lobe, and just how connected all the systems are, especially in the eye. That's all I have.